Welcome to Virology Live. This is session number 15. Today we're going to talk about mechanisms of pathogenesis. What is pathogenesis? Genesis, the development of, right? You can get that word. Patho, damage. The processes that lead to disease. That is what we mean when we say pathogenesis. Now, as we've discussed before, the outcome, the development of disease, is often a result of the parasitic nature of viruses, right? They take many things from the host because most of what they require to reproduce, they cannot provide. So viruses take from the host, and in so doing, they damage them. And so it is just what happens. It is not planned or needed or necessary. And as you'll see today, some viruses do not damage the host cell. But I want to mention at the outset, and we'll return to this later when we talk about evolution, the selective pressures that control evolution of viruses act only on their abilities to reproduce. That is my professional opinion. A lot of things in science are opinions, as you know, and they should be based on facts. And my opinion has been, from studying viruses for nearly 40 years, is that the selection is at the level of making it possible for a virus to reproduce, or maybe increasing that efficiency. It is not to make viruses cause more damage. There is no selective forces that would make a virus virulent and more virulent unless it is to increase reproduction. The primary selection would be reproduction, and perhaps a corollary effect is an increase in virulence. That I would say, yes, that would make sense. But to say that there is selection for greater virulence per se is simply incorrect, and I don't care who you are and how much evolutionary biology you have studied. It makes zero sense because you really have to understand what drives viral evolution to make that statement. So reproduction, and of course that includes finding new hosts as well, because if a virus doesn't find a new host, it's out of business. Now, when we study pathogenesis, we need to use animal models. We cannot study pathogenesis in cells in culture. We can do some things in cells in culture to understand mechanisms, but in the end, if you want to understand the development of disease, you need to study an animal, right? Because disease is, by definition, what happens in an animal. When in a cell, it's killing the cell, cytopathic effects. And so we use animal models, and the mantra here, of course, is mice lie and monkeys exaggerate. It's an old adage that virologists use. And what does that mean? Well, no animal model predicts what is going to happen in people. End of story. I don't care how much you love your animal model. It does not predict what happens in people. I was on a panel many years ago at the New York Academy of Sciences. It was a debate about working on avian H5N1 influenza viruses. And I said this. And Laurie Garrett was on the panel. And she said, well, then I don't know why we use them at all. Well, we use them because they provide some information about mechanisms of pathogenesis, which we can then look into people. We can't do challenges for the most part, but we can look in people and see if some of it makes sense. We can make measurements that would make sense. So just remember that. Mice lie and monkeys exaggerate. What does the monkeys exaggerate mean? Well, chimps are 99% human as far as genetic material goes, but often they have completely different responses to infection. For example, Peter Palese always used to tell me, Peter Palese was my PhD advisor. He used to say, in a non-human primate, if you put influenza virus in the nose, doesn't infect very well. You have to put it way down into the trachea, all right? And we, of course, don't have to do that in people. So Monkeys lie, mice lie, monkeys exaggerate. And then there's an extension of that, which is ferrets are not people. You can actually buy a button over at Cafe Press that says that. One of our listeners made that years ago. All right, so let's talk about how we would use mice or mouse models to study virus infection. So here's our mouse here in the middle, and you want to use it to study virus infection. So you can do a lot of things. You can use, in some cases, human viruses will actually infect mice without any manipulation of the virus or the mouse. But that's not always the case. So sometimes you need to study animal viruses that model human infection. So a virus of mice, it's not a human virus, but it infects mice, causes a similar disease. You can learn a lot of things. Lymphocytic choroidal meningitis virus is a rodent virus that's been used extensively in immunological studies, and therefore can be used to provide a lot of information. In fact, we'll look at some studies with that virus today. Now, the other things you can do in mice, I had a thought there and it went away, unfortunately. Maybe it'll come back. On the left are some of the things that you can do to the mice to enhance your studies. You can modulate the immune response. For example, you can delete immune aspects. You can take out interferon systems. You can take out T cells, B cells, NK cells, right? You can do specific immune cell depletions. You can even introduce clonal T cell receptors, T cells that recognize one peptide, and see what happens. So those are very useful to understand what parts of the immune system are important for studying a disease, right? And then if you say, well, NK cells are important, then you can look in people and see if that's the case. You can't delete them in people, but you can make observations. So that's the key to linking the results of animal studies with people. The animal studies never tell you the whole story. And if you think they do, well, you're just wrong. I'm sorry. You can put human xenografts. What is that? You take a human tissue and you implant it into the mouse. And typically you have to use a nude mouse, which means there's no immune system in the mouse. Otherwise, the tissue would be immediately rejected. And then you can infect. For example, you can make mice somewhat susceptible to HIV by implanting a human immune system, replacing what you've taken out. And you can use other tissues as well. And then you can modulate susceptibility or permissivity. Right? Remember what those terms mean, right? I hope you still remember. You can produce individual viral genes. You can put complete genomes into mice. This has been done with hepatitis B virus to study pathogenesis. And then you can make transgenic mice that produce human receptors, because maybe that's the only block between a virus infecting a mouse, is that there is no receptor present. So mice are hugely manipulable, more so than, I would say, any other animal species that we work with. They're very easy to breed. They're easy to genetically manipulate. And so we and others use them in very large numbers. Other animals are not inbred. There are a lot of inbred mouse lines. Other animals are not inbred, and they're more difficult to genetically manipulate. So you're limited as to what you can do. Now, the first transgenic animal model for a virus infection was the CD155 transgenic mice. CD155 is also known as the poliovirus receptor. This gene was identified actually in my laboratory at Columbia University many years ago by one of my first students, Kathy Mendelson. She identified the gene, which when you put that gene into mouse cells and culture, it allows them to be infected with poliovirus. And so another student of mine then took the human gene encoding the poliovirus receptor, and made transgenic mice producing it. And these mice now can be infected with poliovirus and they become paralyzed. The first transgenic mouse model for a human disease. And many others have, of course, followed since. So that was a very exciting time in the laboratory. Of course, now we have a number of animal models of COVID-19, or that's the disease, of course, mice and other animals that can be infected with SARS-CoV-2. Now, for SARS-CoV-2, mice are not susceptible to infection. In fact, I showed you a result in the fifth lecture, the session on virus entry, that said that mouse cells could not be infected with SARS-CoV-2. And that's because ACE2 in mice doesn't bind the virus. However, the Barrick lab showed very early on that you could passage a virus from mouse to mouse, and after 3 to 9 passages, you end up with a virus that has 2 amino acid changes in the gene encoding in the spike protein that allow binding to mouse ACE2. And you can introduce those separately and also achieve a virus that can now infect mice because it can bind ACE2. And it's interesting that the permissivity of mouse cells is there already, right? The SARS-CoV-2 it can reproduce in mouse cells as long as the receptor is present. So that's one model. But these mice, when infected with this mouse-adapted virus, so this is called a mouse-adapted virus because you passage it in mice. The virus, you select for changes in the virus that allow reproduction in mice. And this has been done for many, many other viruses. It's nothing new. This virus does not cause severe disease in mice. It doesn't kill them at all. They lose some weight, they get some lung infiltrates, but they survive. And so Ralph's laboratory found that if you infect aged mice, you get a little more severe disease, but it's still not the same as what we see in humans. There have also been ACE2 transgenic made. These were actually made after SARS-CoV outbreak in 2003 to study that virus. And a number of labs were about to put them away when COVID emerged, and so they used them for COVID. So again, human ACE2 transgenic mice can use non-human primates. These are extremely expensive, and it limits the number of animals you can use. It limits the statistical significance of the work. Only a few laboratories can do that, but they are useful. You can use ferrets. You can use hamsters. All of these are appropriate animal models, but none of them develop severe disease as do humans. All right, so that's how we study pathogenesis. Now, one of the passaging, all right, so someone wants to know what passaging is. So you take the virus, you infect this mouse, and you let it incubate and reproduce in the mice for however many days. And because the virus doesn't grow very well in mice, what you're hoping is that there is a minority population of viruses that can reproduce. And so say after 7 days, you harvest the organs, you amplify whatever virus is there, and then you infect new mice. And then you do the same thing. So mouse-to-mouse infection is basically passage. You can do the same thing in cell culture. So what do we study with these animal models? One of them is tissue tropism. Very interesting subject. The spectrum of tissues that are infected by a particular virus. Now, all viruses infect every tissue. Some of them are very specific. Some of them are pantropic, right? That is, they infect many tissues, but many have limited tropism. So we call these, according to the tissue that the virus reproduces in, enterotropic. You might guess what that virus, that it infects the enteric tract, the intestines. Neurotropic, viruses that infect neural cells. It's neuro, which means pretty much all the neural cells that are in the CNS. If you want to make it specific to neurons, you would say neuronotropic, right? Hepatotropic, liver cells, etc. And as I said, viruses range from limited, where they only reproduce in a few tissues, like poliovirus, the intestine, and neurons, in fact, to pantropic, where they can infect many tissues. Okay, what are the determinants of tropism? What makes some viruses able to reproduce in many tissues and some limited to a few? Well, here are some of the determinants. Susceptibility, whether or not the receptor is present, right? And obviously, if there's no receptor on a kidney, the virus will not reproduce there, assuming it gets there in the first place, right? So accessibility is a big deal. So if a virus is limited to the respiratory tract, it will not have an opportunity to reach the kidney, especially if there's no viremic phase. So accessibility of the tissues to a virus is important. Permissivity, the internal environment of the cell, which permits it to reproduce in the cell. Immune defenses are another key. For poliovirus, in the transgenic mouse model that I just described to you, if you infect those mice with poliovirus, the virus only reproduces in the brain and spinal cord, actually. It doesn't even reproduce in the gut. The mouse gut is different enough from our guts that the virus does not reproduce there. However, if you remove the interferon defense by simply knocking out the type 1 interferon gene in those transgenic mice, and when you infect them, then the virus reproduces wherever there is a poliovirus receptor, which is pretty much every mouse tissue. Same in humans. So the interferon system controls the tropism of poliovirus. When the interferon system is present, the infection is restricted. When it's absent, the virus can infect many tissues. Now, this is interesting, and how does that apply to people? Well, as you know, only 1 in 100 or 200 people develop paralytic disease. Actually, you may not know that yet. I'm going to tell you that in a subsequent session. And it may be that those individuals have defects in their innate immune system, specifically interferon genes, maybe sensors, maybe signaling proteins, and you can test that. You could go and do a genomic study in people, and some people are doing that, to see if there are people who develop polio have changes in their interferon system genes. So that's how a mouse model is useful. You get fundamental information, then you try and test it in people. And here again, just to remind you that cell receptors are on the cell surface for many viruses, and they can be determinants of tropism. The other factor that can determine tropism is cleavage or proteolytic processing of spike glycoproteins. So here's the HA, the hemagglutinin of influenza viruses, and you've seen this picture before. I know that you won't remember it, but the HA is a membrane protein in the virus particle or on the cell surface, and it has to be cleaved in order to expose the fusion peptide for fusion in the endosome. And the cleavage site occurs where this little orange arrowhead is, and that cleaves the protein, and then when the pH drops, the fusion peptide can be exposed. If it's not cleaved, low pH will do nothing and infection stops. So cleavage is an important determinant of infection. So influenza viruses, for example, are produced in the respiratory tract, and they're cleaved by secreted proteases. And one of them is actually a membrane-bound form is TMPRSS2, transmembrane protease serine 2, which is also involved in cleavage of the SARS-CoV-2 spike protein. And so these can be cleaved on entry in a new cell, or there can be secreted proteases that cleave. And these proteases would be specific to the respiratory tract. So, for example, TMPRSS2 is highly produced in respiratory tissues, but very little elsewhere. And so this is a determinant of tropism. Another example is the avian influenza virus, H5N1. The cleavage site for the HA of most human influenza virus strains is processed by proteases that are restricted to the respiratory tract. But the influenza virus H5N1, which is an avian virus that occasionally spills over into people and causes severe disease. It doesn't transmit very well, so it doesn't cause pandemics, but people are watching it because they're afraid it's quite a virulent virus. Its cleavage site at the N-terminus of the fusion peptide is a furin cleavage site, right? Furin, same cleavage site present in the spike of SARS-CoV-2. And furins are found in many different tissues in humans, as opposed to these respiratory proteases like TMPRSS2. Furins are widely found, and that means that this virus has a broad tropism because of that, because it can be cleaved in many tissues. Now, if this influenza virus, say, somehow got to your liver, If it were cleaved in the respiratory tract, it could get to your liver. It could infect a liver cell, but then the viruses produced there would not have their HA cleaved, so that would be the end of the infection. So the tropism would be limited. So proteases can also control tropism. All right, so let's go to a quiz here. Question number 1. Insertion of multiple basic amino acids at the HA cleavage site allows influenza virus to infect many organs. So the furin cleavage site that I just mentioned has multiple basic amino acids. This means that the blank of the virus has changed. Susceptibility, club cell tryptase, permissivity, tropism, all of the above. And while we are doing that, let's check out some questions here. So it's incorrect to say that viruses evolve to be less virulent. If a virus becomes less virulent, it is most likely a side effect of increased fitness. Well, we don't have any data from humans, all right? Most of the viruses that we have now emerged so long ago that we can't compare what they were like then to now. But we do have some recently emerged viruses like HIV, SARS-CoV-2 that we could look at and see if, in fact, virulence could be reduced. Why would that happen? Well, the standard reason is that it would allow hosts to spread more efficiently, right? Because if you're SARS-1, for example, made most people sick and in the hospital. So there it wasn't very easy to spread. And so if a virus could, in theory, move to that. But we have any data to see that. We're going to talk about more in the evolution lecture. And in fact, we'll look at the Australian experiment with rabbits to address that, but not today. Yeah, killing off your customers is a bad business model. That's what we say. But as I said, there, in my opinion, there are no data to support it. And you could go look in the literature, but I've spent 40 years thinking about it, so I'll save you some time. But I'm just saying that evolution, if a virus became more virulent, it would be some byproduct of some other of selection. It's not going to be selected to be more virulent. That doesn't make any sense. The main selection pressure for viruses is to reproduce better and to then move to another host. And reproducing better can help with moving to another host, right? I heard that a mouse herpes virus is candidate for a 9th human herpes because antibodies are found in 30% of people. Well, mice are 40% of mammals, right? So a lot of people have contact with mice. So I'm not sure that alone is enough to make it a new human virus, but we'll see. Is limited tropism bad for viruses? No, not at all. I didn't say it was bad, did I? In fact, many viruses have limited tropism. The SARS-CoV-2 has a very limited tropism to the upper and lower respiratory tract. I would say it's doing quite well. Defective interferon genes, are they mostly activated in epigenetic, specific, or innate? Well, what I'm talking about is changes in the genes themselves, although certainly epigenetic changes, right, that affect their expression could be involved as well. Yeah, for sure, like histone deacetylases. How do we determine a virus to be recently evolved versus recently discovered? Let's save that for the evolution session, right? We are going to talk a lot about that then. Could virus become adapted to new cells permissively? Yeah, of course. It could by, look, this idea of passaging that we talked about from mouse to mouse, right? The same thing happens early in an outbreak when there's spillover. Say the virus comes from a bat into a human. There's probably some early passage from human to human where the virus isn't well adapted yet. It's never seen a human before. I mean, it's close. Just randomly in the bat over years, viruses have emerged that have right changes to infect people. And then in people, you could select for a virus that's even better. I mean, we have never seen that happen because we don't know until after the fact, right? All right, let's check out this quiz here. What did we do? Yeah, the right answer is tropism, right? The insertion of multiple basic sites changes the tropism because now the multiple basic amino acid cleavage site can be cleaved by proteases that are in many different tissues as opposed to the respiratory tract only, right? So that makes it an expanded tropism, not susceptibility or permissivity. No, permissivity is past the receptor stage, right? All right, another goal of studying pathogenesis, and this is a hot topic, viral virulence. So I'm going to spend some time talking about this. Viral virulence. Boy, has this been botched in the COVID pandemic. I mean, listen, look, I've been studying virulence for a long time, viral virulence. I've been thinking about it a lot. And many of the people who have come into COVID have never studied a virus in their life, and they're publishing papers on virulence, and they really do not. They ought to take this course, for starters. Anyway, viral virulence is the capacity of a virus to cause disease in a host. And we talk about virulent viruses or avirulent or attenuated viruses. So these are viruses that cause much less disease. And there can be gradations in between virulent and avirulent. We make attenuated vaccines, right? We make attenuated viruses in a variety of ways that we'll talk about in the vaccine lecture. But they have reduced virulence. They have reduced capacity to cause disease. And of course, you have to define what you mean by disease, but that's fine. We will do that. So we can quantify virulence in our animal models in a variety of ways. We can simply measure virus titer. For some viruses, we know that the titer is a reflection of virulence. We can measure the mean time to death. That's a good one. If an animal dies quicker, the virus may be more virulent. Mean time to appearance of some signs that you can measure. Remember, mice do not have symptoms, right? Well, they can't tell you about them, so you can never talk about symptoms in any animal model. But people do all the time, but they're wrong. They have to be signs. And that's, this is why nomenclature is important. You have to be consistent in what you're talking about. Mean time to appearance of signs, measurement of fever. Weight loss is a common one in animal models. Measurement of pathological lesions. Many viruses we can do this. SARS-CoV-2, we measure lung lesions. Poliovirus, we can measure lesions in the central nervous system. For HIV, we can measure the reduction of CD4 positive lymphocytes because those are killed by virus infection. Many of these are caused by the immune response, as we have already explored in this course. Many immune responses to viruses cause disease, and we'll explore that more today. We can measure viral virulence. I'm going to give you two examples to show you how we can do that. First, on the left, we have mice infected with two kinds of poliovirus, type 1 and type 2. And we are measuring the number of survivors over time. All right? This is called a Kaplan-Meier plot for those of you who would like the actual name. It's a really simple way to show how many mice are alive at day 5, 6, 7, etc. It's easy to do. You go into the mouse facility, and if you can tell if they're dead, right? Now, we're using this for poliovirus because in mice, it will kill the mice. They will initially get paralyzed, but often you come in and they've already gone through the paralysis, and then they're dead. So we just use the endpoint death. But if you wanted to check them every few hours, you could measure paralysis. So you can see this type 1 virus and these mice are given the same amount of virus by the same route. You have to do it all the same, otherwise you can't compare. And you see type 1, nobody dies. They're all alive, whereas type 2 are all dead by day 11. So that's simply survival. That's one measure of virulence. Here on the right is a more subtle measure, which is measuring what we call a neurovirulence score. And these are viruses that can infect the central nervous system. They're neurotropic. And what you can do is you can infect mice with them, and then you can take out the brain or spinal cord, make sections of it, thin sections, look at them under the microscope. You have to stain them so that you can see the cells, right? Because the tissue doesn't have any color on its own. And then you can measure the amount of cell damage. And pathologists are very good at that. They can see, this is this sort of damage, infiltration, damage of neurons and other cells. But another thing is called perivascular cuffing. It's one of my favorite words. So around a blood vessel, you can have cells coming to it, and that's called perivascular cuffing. And so you can measure that and you put a number on it, and that's what's been done here. So we have a relative neurovirulence score of these different viruses, and each bar is a different region of the CNS. The cortex, the brainstem, the spinal cord, for example. And so you've made sections from different parts. And you can see the Japanese encephalitis virus has a high score in all of these. Yellow fever, two areas higher than the other. Same with West Nile. Lyme-Gat, it's kind of reversed, more in the spinal cord. And then dengue, you're not really doing much at all in the brain. Not really a neurotropic virus, and it's known not to be. So these are all flaviviruses, and you can see within one virus family you can have very different outcomes. And understanding why experimentally is quite interesting. So virulence is a relative property. All right? It is, you can't have a number and say this is the virulence number of this virus, because you would have to define exactly what you're doing, because virulence is influenced by the dose, by the route of infection, by the species that you're using, by the age of the species, by the sex, and other factors that influence susceptibility. It is not correct to compare the virulence of different viruses, right? We do that all the time with SARS-CoV-2 and SARS-1. Not correct. They're different. Well, they're in the same family, but my goodness, unless you're doing an animal experiment, to say in people is hard. I mean, people do it all the time. They use a case fatality ratio to approximate that, but that's not really kosher. If you have similar viruses, you have to assay them in the same way, okay? Similar meaning a coronavirus. I mean, the best thing is to have different variants of SARS-CoV-2. Even comparing SARS-CoV-2 to 1 is a bit dicey because they're so different. But that can be done. But you can't compare coronavirus with poliovirus. That's not happening. If you do, you're wrong. And so measuring virulence in humans is really hard because we cannot control the infection. You can imagine all aspects of the infection are different in humans, even though it's us. We're genetically outbred, so we're all very different. And we'll come back to this in a moment. So here's an example of how virulence depends on the route of inoculation. This is the virus I mentioned earlier, lymphocytic choriomeningitis virus. This is a rodent virus. It rarely infects humans. Only a few. There have been some actually lethal human infections because, let's say, an immunosuppressed person has a pet hamster. The hamster may have LCMV, and then that infects the person. And since their immune system is compromised, they may die. Anyway, here we've inoculated mice two different ways. Intraperitoneally, put a needle in the peritoneum, right, the abdomen. So you're putting virus in the space among the intestines. You're not putting it in the intestine. You're putting it in the space down there, the peritoneum, and then it diffuses and goes to different places. So you can put 100,000 PFU IP into mice, and they'll all survive, right? No problem. But if you put one PFU right in the brain, every one of them dies. So the route of inoculation makes a big deal. Obviously, these viruses have a hard time getting into the brain from the peritoneum. But it just shows you that the route makes a big difference. In my opinion, it is not possible to conclude that one SARS-CoV-2 variant is more virulent than another simply from observations of hospitalization. And you can all disagree with me, but I will say then read the 10 years literature previous to this before you disagree with me. Because why? Well, first of all, it's an observational study. And here's one of them. It's a preprint. I don't know if this will ever get published. It should not. I find these are of limited value in publishing these kinds of observations. Progressive increase in virulence of novel variants in Ontario. This is almost certainly wrong because they are observational studies, which means there are no control groups, and the populations are often different. So you compare the hospitalization now with hospitalization a year ago, it's a totally different population. You have no idea how much virus these people have acquired. So completely uncontrolled experiment. And I can tell you that you can find other preprints which say the opposite of this, that there's no increase in virulence. So these are, in my opinion, nearly useless studies because they tell you nothing mechanistically. They're almost always wrong. And in the end, they're not going to help us in any way with public health measures. If you want to study this, why a variant might be more or less virulent, use an animal model. And, in the course of a pandemic, it takes time to do that. Now, a major goal when we study virulence is to identify the viral and host genes that regulate it, that determine whether a virus is virulent or not. And usually we do this by mutation in a controlled situation, right? So if you have two variants that differ by one amino acid, they usually don't. They usually differ by 10 or more. You have to take them one amino acid at a time and introduce them and ask what's their effect in an animal model. It's something you simply can't do in people. And so what we do in animals is we introduce mutations into the genome, either because we've seen them in people and viruses infecting people, or we have an idea that this particular gene is important. And then we ask, does the mutation affect disease in our animal model? Okay? So here's an example of an experiment to identify virulence genes in a virus. So here you're starting with a virus, and you say, what makes this virus virulent? So you have the wild-type virus. You inject it intracerebrally into mice, and then it will reproduce in the brain, and it will paralyze the mice or do some damage to the CNS that you can measure. Right? So we call this a neurovirulent virus. In cells and culture, it grows very well. These are nice big plaques representative of the fact that the virus is growing really well. So then let's say you take this virus and you introduce mutations throughout the genome or in a protein, in the gene encoding a protein that you think is important. And you identify one which you inject into the mouse, and it doesn't cause disease. But at the same time, it doesn't reproduce well in the mouse or in culture. So this is not very interesting. This is a mutation that has led to a general defect in the ability of the virus to reproduce. All right? It is not a virulence gene per se. It does not affect virulence specifically. It simply affects the ability of the virus to grow in cells. And we're not interested in that. But here's an interesting change. This is a mutation that reduces virulence in mice. So now we call it an attenuated virus. It reproduces poorly in mice, but it grows just fine in cells and culture. So that is a gene specifically required for virulence, and that's the kind of gene we're interested in. Well, you could be interested in genes that affect reproduction overall. That's fine. You make a change in an RNA polymerase. Yeah, the virus doesn't grow well. How interesting is that? But here's a gene that specifically affects disease. That's interesting, and then we want to know how that works. And over the years, many people have done this. It's been going on for as long as virology has been a modern science. And in all of those studies, you can identify 4 classes of what I would say are viral virulence genes, which means they're needed for producing disease in an animal. And these are genes that affect virus replication. All right, those are the ones that are less interesting. There are genes that encode toxins. This is pretty rare for viruses, but I'll give you an example. And the third one is the biggest, genes encoding modifiers of host defense mechanisms. So the antagonism genes that we've talked about last week that antagonize every step of the innate and adaptive responses, if you remove them, they make it difficult for the virus to reproduce because it's eliminated by the immune system. And then finally, genes that encode proteins that enable the virus to spread in the host from one tissue to another can affect virulence. Now, you can make changes in these last two. They will have no impact on reproduction in cell culture because, first, there's no immune system and in cells in culture, and there's no spread involved from, say, the respiratory tract to the skin. We don't have that in the cell culture. So those viruses reproduce just fine in cell culture. And for years, people would call them non-essential genes. They're just studying them in cell culture. Oh, we took out this E4 gene of adenovirus, and the virus is growing just fine. It must be non-essential. No, it's essential somewhere. It's essential for reproduction in animals, which is where this virus arose, not in cells in culture. It's a bad name to call them non-essential genes. I have to say, my job at Columbia, my one and only job at the professor level, I've been there since 1982. I was hired by Harry Ginsberg. He was the chair of my department. He was an adenovirologist. And he would always give seminars and saying, there's no such thing as a non-essential gene. It's needed for something. We just don't know for what. And I always, at the beginning, I thought, what is he, cranky about this? And then I realized he was right. You listen and learn, right? Now, virulence determinants don't have to encode proteins. For example, the vaccine of strains of poliovirus that were produced by Albert Sabin, we're going to talk about these later on. We are going to talk about how he made them and what the changes are. Each of those three strains or serotypes have a change in the 5' non-coding region that reduces neurovirulence. So here's the poliovirus genome. Here is the 5' non-coding region, which is the region before the part of the genome that encodes protein, right? And it's all structured as we have seen before. It's an internal ribosome entry site. And the three Sabin vaccine strains, each has a different single base change in stem loop 5 right here. Here's stem loop 5 expanded. Type 1, type 2, and type 3. He separately selected all of these, and he selected them for reduced virulence in his animal model. And they all turn out to have a single base change in the 5' non-coding region. And these are really important in reducing the virulence of those viruses. And I'll show you an experiment to show that. This was done in my lab. I studied with my students and postdocs for many years what controls the virulence of poliovirus. So here's an experiment where we have taken two polioviruses that differ by a single base at 472. I'll go back to this slide. 472 is right here. It's in the Sabin vaccine, there's a U here, and in the virulent, it's the type 3 vaccine, and in the virulent version, there's a C. So we made two viruses with either a U or a C at that position, and then we injected them into our transgenic mice, and we measure two different things. Here on the top, we're measuring the dose of virus that kills half of the animals. We call that the lethal dose 50%. All right? You can't kill half of the mice when you inject this virus in. You could put 2 × 10⁷ PFU, which is as much as we can put in, and nothing happens. The mice are fine. So that single base makes a big difference. And here's the control. The C at that position, 9,000 PFU inoculated into the brain of mice will paralyze or will kill half of them. Amazing. A single base change in a non-coding region. And here's a growth curve of the virus in the brain of those mice. So we're looking at PFU per gram of tissue on the y-axis and the day after infection on the x-axis. And here's the virus with a C in red. It grows really well. By day 5, it's peaked, okay, and it's associated with paralysis. But the other virus with the U, the titer is not really increasing, and it's declining and eventually cleared. So a single base change does this, and we still to this day do not understand how that works. And we never will, because at some point, types 2 and 3 polio are eradicated. We can't work with them anymore, and type 1 is about to be. But it's an interesting example. There are many viral gene products that modulate host defenses. We've talked about these already, but just to remind you, There are all kinds of immune modulators. There are proteins that modulate apoptosis, autophagy. There are intrinsic proteins that modify intrinsic defenses, like ApoBec3G is modified by a viral protein that sends it for degradation. There are virokines and viroceptors. These are viral homologs of chemokines and chemokine receptors. So these are decoy proteins. So the virus makes a chemokine homolog that binds the receptor but doesn't do anything. So it basically nullifies the immune response. There are complement binding proteins that antagonize that pathway. There are modifiers of MHC1 and 2, as you saw last week. So these are not required for growth in cell culture. Quite frequently, they're just needed to make disease in animals. And if you take them out, you have attenuated disease. So let's look at an example of that. Here is a gamma herpes virus 68. It's a murine herpes virus that's often studied because it infects mice in the laboratory. This is a specific gene called M3. The M3 gene encodes a chemokine receptor. So this is a soluble protein that is produced from virus-infected cells that will bind up chemokines that are produced by the host immune response, and then the chemokines can't work. Chemokines would be attracting immune cells. Now they don't because they're bound up to this fake chemokine receptor. All right, so this is an experiment where we're inoculating mice intracerebrally, and then the mice die. You can also take out the brain and look at cellular infiltrates, immune cells moving into the brain, but we're just going to look at lethality. So we're looking at percent survival here with dose of virus. So let's start with the wild-type virus in blue. The 50% dose, it's a nice way to compare them, is right here. Looks like it's about 5, right? At least halfway between 1 and 10. This is a log scale. So right there is the 50% dose for wild-type virus. Now, we have deleted the M3 gene from the green virus, delta M3. So now the 50% dose, look at this, it's over 100 PFU. So the removal of that gene has attenuated the virulence of this virus because it's an immune antagonist. It's required for reproduction. This is a virus made by genetic manipulation, and to make sure that we haven't inadvertently modified some other part of the genome, which could happen, we put the gene back. We put the M3 gene back, and we test that. That's called M3MR, and that's red. And you can see its virulence is now equivalent to that of wild type. So the M3 gene, which encodes a soluble chemokine receptor, is needed for virulence of this particular herpes virus. Virulence is measured by death. I promised I'd tell you about some toxins. There are not a lot of viral toxins. The bacterial world is full of toxins, enterotoxins that give you diarrhea and so forth. But very few in the virus world. Here's one. It's a protein produced by rotaviruses. These are viruses that give you gastroenteritis, vomiting and diarrhea. And the virus produces a protein called NSP4, which is a non-structural glycoprotein. What does that mean? It's not in the virus particle. It's only in infected cells. It's actually secreted from infected cells. It's NSP4. So here's the intestinal epithelium, and we have NSP4 out there. It's produced because these cells are infected. This protein actually inhibits what's called the sodium-glucose luminal cotransporter. It's a protein in the membrane that will pump one thing in and another thing out, right? That's why it's called a cotransporter, because they're done at the same time. Sodium and glucose are exchanged. And it inhibits it, and that results in fluid imbalance and causes diarrhea. You can actually make NSP4 and feed it to animals, and they will get diarrhea as a consequence. NSP4 also stimulates intracellular calcium levels, which also contributes to the fluid imbalance. In other words, you can't retain fluid in the cells. It just comes out, and that's part of the diarrhea. So that's a viral toxin. If you take the gene, NSP4 gene, out of the virus, it can reproduce in cells in culture because it's not necessary. But here it is producing diarrhea. Now, you may ask, what's the function of this? I think that part of it is enabling transmission to another host, right? It may have some other roles in reproduction in the animal, but I think having diarrhea facilitates transmission. All right, time for another quiz question. Which statement about viral virulence is wrong? It can be influenced by dose, route of infection, species, age, gender, and susceptibility of the host. It can be quantitated by measurement of fever. Ebola virus is more virulent than human papillomavirus. It's the capacity of a virus to cause disease in the host. When comparing virulence, the assays must be the same. All right, take a crack at that, and I'll take a crack at your questions. I forgot to mention that I marked the last one. Okay, here we go. How tight is reproduction and virulence linked? I don't know what to tell you about how tight they are linked, but there haven't been a lot of studies because it's difficult to measure, to alter reproduction. All we know is that if you make a virus that for some reason reproduces to 10 times lower level, it's likely to be attenuated in the host, but it depends on the virus. Someone's complaining that I don't read the entire question. Don't I read the entire question? I do read the entire question, and I read the answers. I don't know what I'm doing that you don't like. Sorry. Is it not the viral cytopathic effect that's of pathophysiological significance? Actually, it's not. It is the immune response. Moderate disease, there is reproduction in the upper tract, but all your symptoms, and cells are killed, but all your symptoms are immune-mediated, right? The coughing, the sneezing, etc. And then if you go on to late severe inflammatory disease, it's all immune-mediated. So killing the viral cytopathic effect has very little to do with that. At best, it's going to stimulate an innate response, which, right, it then gives you good adaptive responses. Remember, cytopathic viruses have good adaptive responses. Why is comparing CFR severe disease from different viruses wrong? I just explained it to you. Because they're different viruses. You can't compare them. Different assay systems, different amounts of virus. Anyway, CFR is wrong because, as I told you earlier, CFR is not an intrinsic measure of viral virulence. It is influenced by the healthcare system. I showed you a graph in the very beginning of this lecture, how it changes during an outbreak. And I found a great graph last night which shows the CFR in the last year and how it was very high early on, and now in most countries that have been vaccinated, it's low. Why? Because we're vaccinating. So you can't use CFR. It's too influenced by interventions. And, folks, I'm a little offended if you refer to me as he. Talk to me, okay? Because I'm here talking to you. So don't say he's doing this or he's doing that. Put a question to me. Professor, please don't do this or that. Thank you. Is CRISPR poised to enhance older vaccines for different diseases? No, not yet. It's not going to do that. It's going to be involved in perhaps modifying host genes, but not vaccines. Statisticians can usually adjust for sex, age, No, not always. How can you adjust for looking at a different hospital population now versus a year ago? You can do sex and age, yeah, but there's a limit to what they can do. In the end, virulence is a biological question, not an epidemiological question. It cannot be assessed by epidemiological studies, and that's part of the problem we have now. People think it can be. So if one can't compare virulence, could you explain the rationale? The rationale is these viruses on their own have a certain high virulence. We don't have interventions, and they can, well, they don't all transmit well. So on their own, they're virulent. They're not comparing them. Virulence is the capacity to cause disease. It was on that slide. Virulence covers more than... Yes, of course it does. I showed you a slide of all the things we can measure. Yeah, non-essential genes is like junk DNA. You don't know what it does, therefore it does nothing. Yes, exactly right. Exactly right. Could one make less virulent viruses that reproduce better than wild type? I think that would be hard, but the Sabin vaccine strains of poliovirus, which are attenuated, they reproduce really well. Not quite as well as the wild type, but enough to give you a good immune response. Making it higher would be difficult. Any correlation between virulence and a subject's existing health? Of course. Yes. People can have underlying genetic defects that make a virus more virulent in them. That's what I think is going on with the 1 in 100 people that get paralyzed by polio. In most people, it's just a inconsequential gut infection, right? But some people get paralyzed. That's not a virus issue. That's a host issue for sure. There's no selection for poliovirus paralyzing people, right? The virus goes in the brain. It's a dead end. It can't spread to another person. It's an accident. So there's some host issue there for sure. And so if you're in poor health, as we'll see, if you're undernourished, viruses are going to be more virulent in you. In respiratory viruses, can the number of particles inhaled relate to virulence? Well, it could. First of all, in people, we don't know because we can never know how much a person's been infected with. Now, there are challenge experiments going on, but they're not going to get to the point of high virulence because they don't want to kill the people. In the lab, you can do this. You can give animals more disease. And what happens? You can give animals more virus. And what happens, they more rapidly progress to disease. But I'm not aware where a disease is fundamentally changed by the inoculum. Could the virus have different virulence in different people? Sure, because people are genetically different, right? They also have different, they have different immune systems, they eat differently, they have different nourishment, etc. Yeah, absolutely. And that's the problem with people because we're all different from each other genetically. Absolutely. Happy Thanksgiving. Thank you so much. I appreciate it. Okay, we're not going to talk about that one. Are investigators working on investigating which part of SARS-CoV-2? Yeah, they are, but not in people, right? Because you can't do that, but they're doing it in experimental animals. And then we can look in people and say, are these similar changes affecting what we observe in people? It's the best that you can do. But yes, it's going on, but it takes a long time, and that's why we don't see them yet. If the disease from SARS-CoV-2 is from the immune response, would not immunosuppression obviate serious illness? Yes, that's why we give dexamethasone in the late inflammatory stage. Not early, because you don't want to suppress the immune response early. Otherwise, you don't clear the virus, right? There is a peak of virus production. It goes down within a week. And then, only then do you get the late inflammatory disease. And that's when you can treat people with dex. Not before then. Didn't Ralph Barrick's lab study virulence of one of the variants with hamsters? Yes. And so that many people are looking at these variants of concern in different animal models. And I think you have to look at more than one report, and you have to look at the magnitude of the difference. If there's a twofold difference, is that going to mean anything in people? Very hard to extrapolate in people. The virulent phenotype is related to the ability of the virus to continue to reproduce. Well, yes, of course. If a virus is not reproducing, it can't express its virulence, right? So reproduction is associated with virulence, yes. If you simply reduce the reproductive capacity of a virus, that makes it less virulent, but it's not from an interesting change, as I was talking about before. you're telling why do I have to get criticized for saying that epidemiologists are wrong? It's a fact. They are good in what they do, but they cannot tell you about the virulence of a virus. You're telling epis they need to communicate, they need to use... All right, so tell me how virulence is going to have any impact on public health measures. You should get vaccinated, you should wear a mask, you should distance. And first of all, transmissibility is a joke because you can't figure that out by population studies like they're doing. So that's why I'm saying take what they say with a grain of salt. I'm not telling them to stay off my turf, although you cannot tell me if a virus is more virulent or not. That needs to be done in a laboratory. So stating that a virus is more virulent is an incomplete statement. It needs additions to define under what condition. Bingo, Franz, you got it. You have to tell me the assay. And so that's why a hospital in Ontario versus a hospital in Scotland comparing hospitalization rates is ridiculous. It's ridiculous. I'm just a stupid guy who enjoys learning. No, you're not stupid. If you're here, you're not stupid. I've listened long enough to trust you. You don't have to trust me. I usually will give you the scientific basis for what I say. Most of your twibs are over my head. I'm sorry. I'm sorry they're over your head. I think if you keep listening, and especially if you take this course, you're going to get a good background. All right, let's go back to the quiz. Could spend all day just answering questions, right? Which is wrong. Ebola is more virulent than a human papilloma. You can't compare different viruses. What do I mean? Different families. Ebola virus is one family, polio is another. They have different routes of inoculation, different animal models. You can't compare them. You could say Ebola virus is virulent for sure because it kills people. You could give me a CFR, but don't compare it to another virus. Okay, that's all. Everything else is... virulence can be quantitated by fever, sure. That's fine. A lot of you pick that, but it is actually okay. All right. Let's look at some cellular virulence determinants. We've looked at viral determinants so far, right? How about some cellular ones? Here's herpes simplex virus. So normally, herpes simplex virus, you get a primary infection. We're going to talk about this, I think, next week. You get a primary infection in your oral mucosa, respiratory mucosa. The virus goes latent in peripheral nervous system. These are the neurons outside of the spinal cord and brain. And periodically, if you get stressed, it reactivates. The virus starts to reproduce. You get a cold sore, right? In very rare cases, the virus goes the wrong way. It goes into the central nervous system to the brain, and it causes encephalitis. Encephalitis, inflammation of the brain. And this is a very rare infection. It's one in about 250,000 people a year who have herpes. And it can be fatal, although it can be treated with antivirals. 70% mortality if untreated. And there are two peaks of incidence between 6 months of age and 3 years. And kids, that's the primary infection. That's when you first get herpes simplex from usually your parents when they kiss you. And the primary infection is also associated at a very low rate with encephalitis. And then over 50 years of age, there's another peak of encephalitis incidence, which is associated with reactivation. So this is actually a great example of how epidemiology and basic virology can work together, right? The epidemiologists tell us when are the peaks of infection, and we explain it biologically. That's the great division of labor. And I'm not trying to exclude anyone. Just stay within your bounds, right? I don't figure out these peaks of infection. I let them do. Anyway, why is this? Why do 1 in 250,000 people develop herpes simplex encephalitis? Well, it's because they are genetically different. What we can do is called GWAS, genome-wide association studies. You sequence the genome of people who develop encephalitis and those who do not, and you look for single nucleotide polymorphisms, which we call SNPs, that associate with encephalitis. This is not easy to do because every human's genome is different from everyone else's. So there's a lot of noise. So you have to somehow get through the noise, which takes a lot of computational work. And but you can do it if you have a rare enough disease and you have a good patient population. So this has been done for herpes simplex encephalitis. And in fact, there's a very well-known group at the Rockefeller here in New York, headed by Jean-Laurent Casanova, who does this for many different diseases, does GWAS, looking for SNPs. By the way, they're called SNPs, not mutations, because who's to say it's a mutation? What's wild type? It's just a change. It's just a difference. And they find changes in genes, these genes, TLR3, UNC93B, TRIF, or TRAF3. They predispose people to encephalitis. And here's a case where you can not only look at the epidemiology and say, yeah, if you have these changes, you're more likely to get encephalitis, but you can take cells from those patients in culture. You can show that herpes reproduces really much better in them than in wild-type cells, that is without the SNP. And if you correct it, then viral loads decrease. So you can do in vitro experiments to support this. So you may want to know, what are these genes? Well, you know what TLR3 is, right? Toll-like receptor 3. It's a sensor in the endosome. And UNC93B is actually a protein in the ER that helps TLR3 get to the endosome. And TRIF and TRAF are parts of the signaling pathways downstream of TLR3. That means, TLR3 senses some viral nucleic acid, and then TRIF and TRAF push signals to the nucleus and then turn on the production of cytokines. And so these individuals have defects in these proteins that make them work less well, so the virus can reproduce really well, and maybe that's a factor in it going from the peripheral nervous system into the brain eventually. And so this is beautiful. This is gorgeous work. It tells you why some people develop different disease than others. We're all different. And so polio, it's going to be the same thing. The 1 in 100 that develop paralysis is going to be linked to something like this, and we don't know what it is yet. Now, this has been done for COVID because, as you know, COVID can be mild. It can be asymptomatic, mild, intermediate, whatever, to severe. So this particular consortium, Genetics of Mortality in Critical Care, it's a GWAS in 2,200 critically ill patients from 208 UK ICUs. So these are really sick people. They have to be in the ICU, which means they can't breathe. They're probably getting high-flow oxygen. Most of them are going to die. And so how are they different from other people? So they did their genome sequence. They did GWAS, genome-wide association studies. And here's a figure that represents all that data. So this is a probability. It's called a Manhattan plot, actually, because the mutations that stick out look like skyscrapers above the background, right? So here is each chromosome, up to 22. So in each bar, these are all the differences they found between the critical population and the healthy population. And you can see here on chromosome 3, look, there are some changes in genes that are really more frequent in these critical population than in others. And these are involved, these CCRs are chemokine receptors. Look at that. OAS is an interferon-stimulated gene. IFNAR2 is the interferon receptor, type 2 interferon receptor. Type 2 is one of those kinases that are involved in signaling from sensors down into the nucleus. So these are all immune response genes or interferon-stimulated genes. And so these probably predispose the people to severe COVID. And in fact, the ones involved in innate responses, probably you don't make a good innate response, the virus gets out of control early, and that leads to the late hyperinflammatory disease. And here I want to show you the ABO blood group genes are there, and they don't rise above. Here, this red line is above which the change is significant, and that was an early association that turned out to be wrong. And here are the data that show, at least in this population, right? You could go and do this elsewhere, and it should be validated elsewhere, and it's being done. But it's an example of what you can do. It's really, really amazing. There are some host genes that determine susceptibility, and I'm talking about receptors. Here is the CCR5 gene which encodes a chemokine receptor that's needed for HIV entry. Remember, HIV glycoprotein, the yellow and the green protein here, binds CD4 and then a chemokine receptor gene, CCR5 or CXCR4. There's a deletion in the CCR5 gene, which is present in 4 to 16% of people of European descent. These people don't make CCR5. They are protected against HIV infection. And we don't know why this fraction of the population has this deletion. They're fine otherwise. So obviously CCR5 is redundant, right? But it does protect them against HIV infection. And in fact, you have heard, I'm sure, the story of first the German AIDS patient and then the London AIDS patients who received as therapy, they had AIDS, they received a bone marrow transplant from a donor who had the CCR5 delta 32 mutation. So what you do is you ablate the bone marrow of these patients. So now they can't make any of their new immune cells. And then you give them a transplant of bone marrow from a donor who is CCR5 delta 32. And then that bone marrow will populate your immune system. So now all your CD4 cells, which of course are the targets of HIV infection, they no longer have CCR5, so they're resistant to infection. So this cured the AIDS patient and the London patient as well. However, stem cell transplantation is not a solution because it's really expensive and has a high mortality rate associated with it. But it tells us if we could get rid of CCR5 in an AIDS patient, say by CRISPR, we could think about curing it. So we're going to talk about that later on. Other determinants of virulence. Age. Yeah. The very young and the very old are the most susceptible to disease in general. Right? There's always exceptions. So here's a chart of the death rate from influenza between 1911 and 1915 according to age group. And you see the very young and the elderly, not old. I mean, these are 65 and up. Elderly, advanced age, are the most susceptible. If you actually looked at 1918 to 1920, there would be an additional peak of susceptibility here in the middle that we don't actually understand. Anyway, the young, their immune response is immature. Probably explains in part why younger kids don't have as much severe COVID as older. But of course, the older individuals, well, their immune system is senescing. First of all, the muscles in their chest are getting weaker, so they can't expel things as well. The alveoli are less elastic, so they can't get rid of things. There's a diminished cough reflex. The bone marrow diminishes with age. I like to say, my long bones have very little marrow left in them by my age. I am a shell of my former self. So it's hard for you to make new immune cells because you have fewer and fewer. So you're more susceptible to infectious diseases. This also is reflected in COVID. This is a graph which is no longer available at Our World in Data, but it is somewhat instructive. It's the case fatality ratio by age. This was, about 10 months ago. And you can see stratified by age. And as age increases, it goes up in general. These are just for a few different countries. And I showed you this earlier for the first 40,000 cases in China. So age has a big influence, and exactly why is being studied for sure. Sex is another determinant of virulence. It's sex, not gender, all right? Gender is a cultural thing. Sex is a biological difference, and in general, men are slightly more susceptible to infections than females, but not always. It depends on the particular virus and the study. Here's one study, which is referenced down here, where they looked at immune responses to influenza vaccination. So that's a great opportunity to do this kind of study, right? You are vaccinating people against influenza, and you can easily take blood and measure the antibody response. And so here is neutralizing antibody seroconversion rate %. And so we have adult males and adult females. And you can see the female rate is higher. than the males. But when you look at aged males and aged females, so I would be aged, like an aged wine, the difference is gone. And so it's thought, and in fact, this difference, females versus males, is thought to have been selected to ensure reproductive success, right? The female is going to bear the offspring, and so they should be protected against infectious disease. And these responses, at least in these studies, they correlate with elevated estradiol. But, the exact mechanisms are not understood, but they're working on it to figure it out. Also, pregnancy is a big problem for certain infections. Hepatitis A, hepatitis B, hepatitis E, influenza, COVID-19 are all more lethal in pregnant women. Some other determinants of susceptibility. Again, we're talking about host determinants of susceptibility. Malnutrition. Someone asked about this earlier. Absolutely. Malnutrition increases susceptibility because physical barriers and immune responses are compromised. Measles is 300 times more lethal in developing countries than in Europe and North America, mainly because of malnutrition in developing countries. Can you imagine? 300 times more susceptible to measles just by being undernourished. Cigarette smoking is another one. It increases susceptibility to respiratory infections. Not only does it compromise the mucosal barrier, but it also causes changes in innate sensors at those regions. Air pollution, also, areas with higher air pollution. These are obviously difficult studies because hard to tease out other factors that may be affecting susceptibility, but air pollution has been named as another. And stress. If you are stressed for whatever reason, it increases your susceptibility to infectious diseases. Okay, let's go to our last question here. Which statement about determinants of viral virulence is incorrect? A, virulence genes can encode viral proteins. B, virulence genes can encode cellular proteins. C, they are the same in all viruses. They can be found in untranslated regions. They may encode immune modulators. Which one is wrong? Okay, and let us go back to your always scintillating and engaging questions. And I'm not being sarcastic. Would late effects be attributed to virulence? I'm not sure. I mean, you could have an early difference in virulence, which would, say, lead to greater reproduction. That could lead to inflammatory disease. So, yes. Epidemiologists fabricate with educated guess models, but do not explain this to the public. Yes, most of what epidemiologists do is modeling. Sometimes they're right, sometimes they're wrong. And there is a place for that. But yes, many people do not understand it. And so when an epidemiological study concludes that this variant is more virulent, the people do not understand the nuances. And I highly object to that because, as I've told you today, it's often not so conclusive. In Australia, they keep people at home. Well, that's going to make these kinds of in-hospital studies impossible, right? Yeah. I think these hospitalization rates numbers reported by health authorities, mostly for public attention getting, which is why I wonder still what exactly was done in India to keep numbers down. I think that might be part of it. Maybe they want people to distance or mask or get vaccinated. Maybe that's part of it. But from a scientific viewpoint, they're often not accurate, yeah. Can viruses acquire genes of immune signaling molecules during their infection cycle, which then mutate and act as immune suppressors? Well, not in real time today. This is in geological time we're talking about, right? So where these genes originated is a good question. So the viruses that have cytokine homologs, did they originate in the cell? Probably, and then the viruses took them, but a long time ago. And we can track that by phylogenetics. But, in you, it's not going to happen in a compressed period of time. listening to TWIV is like learning a new language. The more you listen, the more it makes sense. Yes, that's a good way to look at it. And people have said the more they listen, the more they get it. Because yes, even though we try hard to explain, we still have a different language and we slip into it. But that's a good way. The more the better. And I understand you don't all have time to do this. I totally get it. Take what you can in drips and drabs. I'm here for the long run, not going anywhere. You can go away and come back. I'll still be here. I got about 20 years, I figure. The problem is that science uses the same words as the general public for different reasons. Yeah. So I think if you want to learn science, you need to learn their lingo, how they use the words. Otherwise, it's, mutation is an easy one, which is completely misused, not only by the public, but by also many scientists. It's not an amino acid change. Why does my MD not know this? Well, MDs are another story, and I don't want to trash MDs. My dad was an MD. He was a surgeon. He knew his things. He knew nothing about infectious diseases. He would go, if he had one in his patient, he would give it to a specialist, right? Or a medical doctor, an internist. Doctors are trained to treat patients, to see symptoms and signs, and to treat accordingly. That's it. Underlying stuff they generally don't get at. Unless they're an academic clinical research physician, then they could do that. But most physicians that you're going to encounter, and let me tell you, I can give you stories about my doctors. I go and talk to them about viruses. Forget it. Gastroenterologists, general physicians, very little knowledge. And why is that? Because they get it very briefly in medical school. And I know because I teach medical students. And then they take their boards and they pass, and then that's it. Then they do their thing and they don't have to encounter it anymore. So really different. And they tend to hear the big picture from CDC, or that's percolating through their professional associations, the pulmonary societies or whatever they're called, blood, heart, etc. They put out these missives, and then that's it. Anyway, I don't want to talk about that. You should ask Daniel about that. We'll get him back, and you can ask him. Yes, so they kind of annoyed me that they said I wasn't reading the question, but I find it important to defend myself. Thank you for your support, Lynn. Really appreciate it. Would the GWAS studies pick up differences that need more than a difference? Yeah, it would pick them up, but then testing them would be the key, right? You'd have to do them in combinations, but you could do that for sure. I've shared the German patient story. So Vanity took this course in the spring of this year, and I'm glad she found some new things that she didn't know before. That's great. I have read somewhere that CCR5 predisposes to flu. No, that I haven't heard any convincing evidence for any of these associations. They've had all kinds of explanations. West Nile is another one, but I don't think they're correct. CCR5 in Europe related to plague. That was an original hypothesis, but it doesn't seem to be correct, no. With age, we gain experience and character. I certainly have changed a bit with age, but I've gotten wiser. I've learned more virology, and I think I've learned better how to teach it, especially how to talk about it, right? Why is latency more severe than the primary? Well, that's a good question. The primary is often cleared at the mucosal barriers, and then the virus, goes latent in the peripheral ganglia. And then when it's reactivated, it's deeper, it's closer to the central nervous system, so it's easy to go the wrong way. But I don't think we really have a good answer. Thank you for your contribution. Really appreciate it. is it not possible after determining a person's genetic code to engineer and administer bone marrow to the age to enable their immune system to retain optimal function? Well, as I said, bone marrow transplants are pretty expensive and they can be lethal, so I'm not sure that that's optimal. But maybe figuring out more specifically what's wrong would be better. What stops regular protein manufacturing in a cell? Why does that not happen with an infection? Well, actually, we talked about that in the infected cell. Not much, but many viruses induce shutoff of host translation, but they do have ways to get their own messages translated. We talked about that for sure. You might need 30 years of teaching. Well, let's see, I'm 68. I'm not going to live to 98. There's no bloody way. No, I'm sorry. I get, see, 20 years, 88, I could do probably. But, you know, if I'm incoherent, you guys are going to tell me, and then it's it. Yeah, MDs can't remember everything they've ever learned. They remember what they deal with, right, often. It's hard. All right. A lot of good questions here. I'll take more of them. With gene therapies, what determines if the fix is persistent? Well, it depends what cell you're trying to do this in, right? If you really want it to be persistent, it has to be in the germline. But because then it's going to be in every cell, right? So, but that's only going to deal with your offspring. So in you have to decide. Stem cells are usually the right place because they're self-renewing and they can make other cells, and so that's the cell you want to target. I don't have an encyclopedic knowledge, but I'm glad you think so. It's always good to think your teacher is knowledgeable. But happy Thanksgiving, and hope to see you some more. You should know your precise scientific learning informs even me. It's important. Thank you. Appreciate it. I really appreciate it. What viruses allow... Oh, wait, first, fungi? Yes, absolutely. There are viruses of fungi. Maybe... So I'm hoping to teach a viruses course next, right? So this course was called Virology Live. The next one will be Viruses Live, and we'll go through different viruses. And maybe we'll include plant and fungal viruses. And that would be... I don't know when I'm going to teach it, but not next semester, because I have to teach this course again at Columbia, so... what mechanisms allow chickenpox and herpes to hide? We're going to talk about that when we talk about persistence. So let's do it next time. All right, let's get back to, this looks like a lot of people are leaving, so let's get back to the course. What's the story here? What's wrong? They're the same in all viruses, that's correct. They can encode viral proteins. Cellular genes can be virulence genes. They can be in untranslated regions. They can encode immune modulators. All that's correct. What is incorrect is that they're the same in all viruses. All right. Actually, we have quite a bit to go now. Mechanisms. How do viruses cause damage to cells? So there's two sections of this. There's direct killing, cytolytic viruses. They cause cytopathic effects like apoptosis, necrosis, pyroptosis. They kill cells. Here, remember the poliovirus time course. Uninfected, 4 hours, 8 hours, 12 hours. The cells round up, detach, and die. Many viruses make viral porins, which are pores that go into the membrane of the cell, and things leak out, so the cells die. Many viruses inhibit host protein synthesis, host RNA synthesis. This causes the membranes to lose integrity. Enzymes leak from lysosomes. It degrades the cytoplasm. Many envelope viruses cause fusion of cells, syncytium formation, which we talked about earlier. These are all different ways that viruses kill cells. And that's certainly a fraction of the pathology of a virus infection. But most of it is immunopathology. Too much of a good thing. Remember inflammation, these clinical signs and symptoms, fever, tissue damage, aches, pains, nausea. Those are mainly a consequence of the host response to infection. You're seeing very little of cytopathic effects when you get SARS-CoV-2 infection. It's mostly host immune response driven. And in fact, for non-cytopathic viruses, the ones that don't kill cells, everything, all the disease is a consequence of the immune response. So let me give you some examples of what we call immunopathology. Here are different kinds of immunopathology, T-cell mediated and B-cell mediated, and of course, we have different kinds of T cells. We have our CD8 positive, our cytotoxic T lymphocytes. These kill infected cells. You can imagine that that's going to cause disease, right? And all these viruses have the disease has a contribution from CD8 positive cells. And how we know this, I'll show you in a moment. CD4 cells, we have two kinds. We have Th1s that produce cytokines that give rise to CTLs or help them mature. And all of these viruses display Th1 positive CD4 mediated immunopathology, respiratory syncytial virus, Th2. And then we have viruses where antibodies cause immunopathologies. So these are all the adaptive responses. There's also overexuberant innate responses, which cause immunopathology as well. All those cytokines and chemokines made in too high levels, not good. So let's look at some experimental data. Here's an example of a virus disease mediated by CD8 positive cytotoxic T lymphocytes. Okay? This is a lymphocytic choriomeningitis virus. We're injecting the virus into the brain of the mouse. That's the mouse virus that I talked about earlier. Within 8 days, the mice are dead. And remember, if you put this virus intraperitoneally, mice would survive. So right in the brain, they die. The virus reproduces in the brain. Now, let's say you inject and then immune suppress the mice. You could give them steroids to broadly suppress the immune response. After 8 days, they're living, but they have a persistent virus infection. The virus is reproducing, hasn't been cleared because you've suppressed the immune response. So that tells you that death is somehow caused by the immune response. Now, if you take CD8 positive CTLs from a mouse that has not been immunosuppressed, but that has been immunized with LCMV, and you give them to these mice, that's called adoptive immunization, within 5 days, they're dead. So you could take all kinds of cells. You could take B cells. You could take CD4s, CD8s, as was done here, macrophages, NK cells, whatever you want to do, and see which one kills them. In this case, it's CD8 cells that's killing the mice. Here's another example of lesions associated with CD8 positive T cells. Again, this is infection of mice with LCMV. So here on the top, we're looking at survival on the upper left, percent alive. And you see the wild-type mice that are infected with LCMV, they're dead within 8 to 10 days, as I told you. But if you knock out the gene encoding perforin, perforin is one of the genes that encodes a protein in cytotoxic T lymphocytes that kills the infected cells. It pokes hole in the infected cell membrane. If you knock out that gene, the mice all survive. CD8-mediated killing is killing the mice too. On the right, we're measuring liver enzymes, so the virus is also causing liver damage. You can see in wild-type mice that this particular liver enzyme goes up in the serum. It shouldn't be in the serum, but when liver cells are damaged, it leaks into the serum. So wild-type mice, lots of liver enzyme in the blood. Perforin knockout, nothing. The virus is not, the CD8s are not causing damage to liver cells because there's no perforin. And finally, we're looking in the heart down here. This is actually a different virus. It's a Coxsackie virus that causes heart damage. Here's wild-type mice. The staining with this blue stain, this is a stain that picks up fibrosis caused by the virus infection. So that's damage. But in a perforin knockout mice, there's no heart damage. So Coxsackie virus is one of the viruses that you can get, and it will destroy your heart, and you need to get a heart transplant as a consequence. But this tells us that modulating the CD8 cells could prevent that in some way. Of course, you need them to clear virus infections, but you can see they can cause damage. CD4 plus lymphocytes are also associated with damage. Of course, CD4s, right, are the ones that provide help. They provide cytokines that help CD8s mature, that help B cells mature. So they make a lot of cytokines. They also recruit and activate many effector cells like neutrophils and mononuclear cells, which in turn can damage cells. And these cells in turn release proteases, reactive radicals like peroxides, cytokines that kill cells like TNF-alpha. So they cause cell damage. So it all starts with the CD4 producing cytokines that attract other cells, which in turn secrete things that damage cells. So let's look at an example of this. Lesions associated with CD4 positive T cells. Herpes stromal keratitis. It's one of the most common causes of blindness in developed countries, because most of us are infected with herpes simplex virus. And if somehow the virus gets to your eye, this can be a real problem. It is an entirely immunopathological disease caused by CD4 positive Th1 cells. What happens here is that if you get repeated infections, your cornea becomes more and more opaque, and you get reduced vision. So what's the basis for this? Well, that's shown here. Here are two sections of the cornea. The cornea, the underlying or the stromal cells are on the bottom here. And then there's an epithelial layer on the top. So two different magnifications. The virus replicates only in the epithelium, the corneal epithelium. And the damage is in the underlying uninfected stromal cells. So there's a layer that makes up the cornea proper, right? There is where the damage occurs and it becomes opaque, but the virus doesn't reproduce there. What happens is the infection in the epithelium recruits CD4 positive T cells to the underlying stroma, and you can see they're darkly staining cells here. The cytokines recruit neutrophils, and then the neutrophils damage the corneal stroma. So the combination of infiltration and inflammatory mediators causes damage, and eventually, with enough infections, blindness. So the virus is not even reproducing in these stromal cells. So this is an entirely immunopathological disease. West Nile virus can cause encephalitis in a certain fraction of individuals, as we've talked about before. And what happens with West Nile virus infection, the infection is sensed by toll-like receptor 3, and TLR3 then turns on the synthesis of cytokines, which include TNF-alpha, which compromises the blood-brain barrier and allows virus to get into the brain, and that's where you get the encephalitis from. And so this can be studied in mice, and that experiment is shown here. For example, we are putting wild-type West Nile virus into either wild-type mice or mice that lack TLR3. So TLR3 null mice are more resistant because they can't sense the virus and they can't make TNF-alpha. So with a wild-type mouse, you infect with West Nile virus. This is day 0, that's the brain. And you also give the mice a blue dye that typically can't get into the brain unless the blood-brain barrier is compromised. So here you see by day 3 after infection, the blood-brain barrier is compromised and the blue dye is getting in the brain. It's quite clear from this picture, right? You use TLR3 null mice. They're lacking the gene for TLR3. You infect them with West Nile virus. Look at day 3, barely any dye in the brain. Because, why is that? They're not making TNF-alpha because they don't have TLR3. So it just shows that the pathology, the encephalitis in this case, is due to the immune response, TNF-alpha compromising the blood-brain barrier. Poxes and rashes are all a combination of virus reproduction and immune responses. We have seen pictures of these before: measles, smallpox, varicella zoster. They all make rashes. These are typically caused by virus reproduction at foci in the skin, and then Th1 cells and macrophages hone in to these sites. They produce cytokines. The cytokines increase capillary permeability. They increase T cells coming into the region, and that's basically what a rash is. It's a focal kind of inflammatory reaction. Now, dengue, or breakbone fever, is an example of a immunopathology based on antibodies. So dengue virus, a flavivirus transmitted mainly by Aedes aegypti. It's endemic in the Caribbean, Central and South America, Africa, Southeast Asia. Many, many billions of people at risk for infection. We see about 400 million infections a year diagnosed, probably many more. It's second only to malaria among insect-borne diseases. And there is a rock group called dengue fever. I think it's a Cambodian rock group. I went into the student radio station at Vanderbilt University once, and they have big racks of CDs, right, for music they play. And this was sitting there on the shelf, and I was going to be interviewed by one of the DJs, and so he played this at the beginning and the end of my interview because I noticed it. Anyway, dengue was almost eradicated before 1981 by the use of DDT to control mosquitoes. You can see prior to 1981, very little laboratory confirmed dengue in Central and South America. But then, of course, we stopped using DDT because it had harmful effects on the ecosystem. And so the infections returned. And one of the reasons they did is that the used tire trade is very good at transporting mosquitoes around the world, right? The tires are picked up all over. They're thrown on container ships and typically uncovered. They fill with water and the mosquitoes breed in them, and they are brought elsewhere. So this is a map of currently where most of the dengue is occurring, equatorial, where most of the Aedes aegypti is, and the dots in the other places like the U.S. are typically imported cases. All right, so the infection, the primary infection, can either be asymptomatic or lead to an acute febrile illness. You get headache, back and limb pain, rash, severe aches and pains in the bones. It's typically self-limiting. You recover in 7 to 10 days. In 1 in 14,000 of these, you can get hemorrhagic fever, where you have capillary hemorrhage and shock, and you can die of that. So internal bleeding can lead to shock syndrome. You make antibodies. There are 4 serotypes. Antibodies against one serotype will not protect against another. All right. You get another infection. Let's say you've got serotype 1 immunity, and then you go somewhere else and you get infected with type 2. Your immune cells, your B cells, make a memory response to the primary serotype, which will bind type 2 in this case, but it will not neutralize it because that's the definition of a serotype. The antibodies don't cross-neutralize. However, the antibody bound to the virus will facilitate its uptake into cells like macrophages, monocytes that have Fc receptors, a receptor on the plasma membrane that will bind the Fc portion of the antibody. The virus will then get into a cell which it would normally not infect. This increases the viral load as a consequence, and it also causes these cells to produce much more cytokines than they normally would. And so this series of events increases the incidence of hemorrhagic fever and shock from 1 in 14,000 in primary infections to 1 in 90 and 1 in 50 in secondary infections. So this is called antibody-dependent enhancement of disease. So it's immunopathology based on B cells. And of course, SARS-CoV infections, whether 1 or 2, have massive amounts of immunopathology involved. We have acute lung injury, acute respiratory distress, the late inflammatory phase of these disease, largely a consequence of dysregulated inflammatory cytokine production, or what is called cytokine storm. And so normally, in an uncomplicated SARS-CoV or CoV-2 infection, You'd have an early effective interferon response, which reduces virus reproduction, inflammatory infiltration of monocytes and neutrophils into the respiratory tract. You'll have cytokines produced, but this will lead to resolution of infection within a week or so. After the peak, the host survives and has protective immunity. But in some individuals, as you know, we have dysregulated inflammation. And so it's the late sequelae of the viral phase. And we think these are a consequence of delayed interferon responses, which then lead to higher virus reproduction, more infiltration of immune cells, more pro-inflammatory cytokine and chemokine production, and then that leads to acute lung injury, acute respiratory distress, and death because of hypoxia. You can't breathe, essentially. You have too much lung damage. So that's an immunopathological response, and as we have said, we're trying to understand what's associated with that. This is one of Daniel Griffin's slides that summarizes these stages. So again, we have the blue is viral load detected by PCR. So you have peak of viral RNA, which is either slightly before or at the time of symptom onset, which is TS. TDVR is detectable viral RNA. TE is the exposure. So, the peak is anywhere from 2 to 14 days after exposure. Symptom onset, and then the RNA loads rapidly decline. Within another 7 days, they are below the infectious level. Yet, as you know, a number of patients go on to this inflammatory period, which is characterized by overexuberant immune responses, decreased lung functions, other issues, multi-system inflammatory disease, and so forth. But these individuals are not contagious for the most part. They have very low viral loads. This is an immune disease. This is why it's called the inflammatory period. And finally, many virus infections on their own cause immunosuppression, which is a global reduction of the immune response caused by infection. This can have different mechanisms. The virus can reproduce in one or more cells of the immune system. The virus can perturb cytokines and signaling in ways that we've talked about. Viral proteins acting as viroreceptors or virokines. This can all perturb the nice harmony that the response has and make it go awry. During measles virus infection, we see immunosuppression. So here's a child with measles, and this study was done by giving the children a tuberculin test where you inoculate some tuberculin antigen intradermally. And then if you have been exposed to TB in the past, you have an infiltration or influx of T cells into this area very quickly, and it forms a raised area called an induration, which can be measured. And so that is what we're looking at here. We're looking at the induration, the size of this response to the tuberculosis vaccine in children. So at baseline, without measles, this is 7 millimeter induration. And then at the peak of rash, look at that. No response to the tuberculin vaccine. A week later, no response. And then finally, 2, 3, and 4 weeks, as the rash is subsiding, then you get the T cell response back. So this is an example of how measles virus infection causes immunosuppression. As you can imagine, this is not good. What's the mechanism? Well, normally when an antigen-presenting cell detects a virus via, say, a toll-like receptor, it makes a certain set of cytokines that either, favor Th1 or Th2 type immunity, right? Th1 cytotoxic T cells. But measles disrupts that. When measles virus is infecting an APC, it turns off the Th1 response and turns on instead a Th2, which favors antibody production, but that's not what you need to recover from measles. And so the T cells are reduced, and that's why you don't, in part, have the homing to the tuberculin site. Now, the other thing that we've learned that measles does, it erases your immune memory. And this was discovered by a group including Michael Minna back in 2019, and we had him on Immune to talk about this. It was before COVID. The virus infects memory B cells and erases any memory that you've had to infection or vaccination. So this paper, that what they did, they took serum from children and they put it on a chip that had the full proteomes of 400 pathogenic human viruses. So they made peptides to all the viral proteins of these 400 viruses. They put them on a glass slide, and you know where each peptide is. So then you put the antibody on top of it from the kid's serum, and you can tell where they're reacting. The chip can be read by a machine, and then you can say, does this child have antibodies to poliovirus or measles or whatever? And here are the results. So normal kids, these are the 2 time points. These kids don't have measles, so there's no difference in their proteomes. See, these little plots are the sum of all the thousands and thousands of peptides against which the serum is reacting. But here are the 2 groups that are informative. These are the mild and severe measles. Look at the decline in the number of reactive peptides from before measles to after measles. So they're losing serum reactivity, antibodies binding to peptides from particular viruses. And they can tell you exactly what viruses are involved by deconvoluting these data. And this is another way of looking at it. This is the proportion of total epitopes detected at time 1 that were retained at time 2, so before and after measles. And so for the control kids, there's no difference, but look at how much measles, mild and severe, goes way down. The number of epitopes recognized by their serum goes way down. So the virus infects memory B cells, erases your immune memory. It'll come back, but you're going to have to be revaccinated, you're going to have to be reinfected, and for a while you're going to be susceptible to infection. So this is another reason why people need to get measles vaccine. So measles virus causes immunosuppression by infecting cells, as we have said, monocytes, dendritic cells, epithelial cells in the thymus, memory B cells. And so you get reduced T cells, you get reduced B cell memory, enhanced infections. Rubella virus also infects lymphoid cells and immunosuppresses. And of course, HIV-1 affects CD4 positive T cells, which you need to make an immune response, right? They make the cytokines that make you make antibodies and T cells, so you die of opportunistic infections that would normally be cleared. So that is immunopathology on Wednesday. Yes, we will have a class Wednesday. Many of you may not be here because of Turkey Day, but that's just a U.S. thing, so all of our international students, I assume you'll be here. We're going to talk about acute infections, infections that come and go in a fixed period of time. We're going to talk about examples of those and how they work. Okay, let's do some questions here. You need some cat viruses? Yeah, if I do a virus course, I can cover all of those things that we normally don't, right? Yeah, so is rabies a form of encephalitis? It has components of encephalitis, but more than that as well. Yes. And so I'm going to do a class on viruses. And then after that, I think I'll do a class on emerging viruses. We'll take a look at all the viruses that have emerged from animal sources over the years. What were the factors, what do they do, and so forth, where they came from. So lots of future virology here. Enjoy the veggie lasagna. Thank you. We're actually going to have, I'm going to make eggplant parm. Is immunopathology generally more common for virus infections versus bacteria? I don't want to answer that. I'm not an expert on bacteria. I don't know the extent to which immunopathology factors in. I'm sure there's part of it, right? Why not? No, this is not the last class. It goes to the last Wednesday I think in December 29th, something like that. So you're stuck for another month, 6 weeks. You have a syllabus. Go check it out. Can I talk about Coxsackie affecting the heart? All right, briefly. Coxsackie virus is one of many viruses that can infect heart tissue, and you don't know you have it. It could be a respiratory acquisition or gut or fecal oral, and then it slowly destroys your heart muscle, and suddenly you have a loss of heart function, and you go get checked out, and they say your heart's destroyed. You need a heart transplant, and you can't pick it up early because you don't have symptoms, and so it's kind of insidious. And that damage is largely immunopathological, as I showed you. I totally get why in earlier sessions you said virulence lethality is mostly an accident. I sort of translate that the virus does not care what happens after it's spread. Yeah, you anthropomorphize, but that's the point. It doesn't matter. It just needs to spread to a new host. And if I look at it as leaving a sinking ship. The ship is sinking because its boiler blew up, right? And the virus just moves on, and the host dies. Yep. So dengue is a pandemic too. So the thing is, pandemic is in part how many countries are involved, and so it's only in those equatorial countries. So it's not called officially a pandemic by WHO. Do I consider the blisters of psoriasis part of an immune response? Well, it's out of my expertise, but yes, it is. Is there any relationship between measles and encephalitis? Yes, measles can cause encephalitis in infected people at a rate of 1 in 1,000, roughly, for sure. Gets into the brain. Is immune enhancement what happened with the kids vaccinated with RSV? No, it was different. The vaccine was treated with formalin, and a number of the important epitopes were destroyed, and so the kids made an immune response, but it was not protective, and so it was more damaging, and I don't think we're going to cover that. I used to, but maybe we can in vaccination, because we now know a lot. After 50 years, we know a lot about it. Is second infection worse with any of the four? I don't think it is, but all four are not always circulating at the same time, so that can also determine whether you get this antibody-dependent enhancement or not. Could viruses which inhibit the interferon response have a fitness advantage, as they could delay the onset, which might lead the host to be more sociable? Yeah, so you're thinking there could be a higher proportion of asymptomatic infections. I mean, theoretically, that would work, right? But then, I don't know if it's going to last very long, because then they have a massive amount of virus, and it leads to the serious sequelae. So it sounds good, but you have to be careful about what we think might work for a virus, right? Glucose intake helpful or detrimental? So glucose, of course, is going to go towards energy production, which viruses require in order to reproduce. So I would say it would be detrimental for a virus infection. Is cytokine storm confirmed? Yeah, it is. They've done cytokine profiles, and there's clearly overproduction of multiple cytokines. Yep. Daniel's slide, the one I showed, well, it's here, but he published that. You could look it up in his paper. What consequence has antibody-mediated enhancement of infection to develop of dengue vaccines? So there is a dengue vaccine called Dengvaxia, which is affected by ADE, yes. So you cannot they decided not to give the vaccine in people who have never been exposed to dengue, because it's looked at as a first infection, and then you can get ADE in the second. So that's a real problem, yeah. when a forced serotype can the immune pathology. So the forced serotype vaccine, the problem with that is the type 2 component is not very good, so that you can have type 2 immunopathology. So there is a better dengue vaccine in development, which is completely protective against all 4 serotypes, and that will solve the problem. It's an attenuated vaccine. How soon can the immune memory come back? Well, if you get it vaccinated, as soon as you recover from the rash, you're making new B cells, so you can get vaccinated right away. So I would say a few weeks after the rash. Yeah. No, memory T cells are not affected by measles. The virus doesn't infect them. Measles erases immune memory for all the pathogens you have memory to. Those peptide studies were all pathogens. It's not specific at all. Could you use B memory cell suppression of measles to fix the secondary dengue? We want to get rid of your dengue memory. I don't think that's. I think you need a good dengue vaccine. That would be the solution. Yeah. I want to thank the moderators today, Vanity. Thank you for being here. Mr. Ozzycam, thank you so much. Kate, thank you for your contribution. Really appreciate it. A number of you will be here. That's great. Also, Steph is here, the moderator, and Les are here as well. Thank you all for moderating. And of course, is this a formal college course? No, it's not. Not at all. This is a live stream YouTube course, which I'm doing for you. And I have a Columbia course that I teach in the spring, which is for them. But I also post the videos on YouTube, so you can watch them again. I've gotten several vaccines as a baby. Did those have mortality side effects? Well, you've got polio vaccine, which could have caused you to be paralyzed. You get measles, which, can cause 1 in 1,000 encephalitis and mortality. You've got chickenpox vaccines. Yeah, so all those viruses cause pretty serious infections and disease. So that's why we have vaccines against them. The more dengue you get, the more pathology. So up to a point, right? Because then you get all 4. If you've got all 4, then you shouldn't get immunopathology, and then that's what a vaccine should do, right? And nitric oxide also plays a role. We didn't talk about that, but nitric oxide is made by an interferon-induced enzyme, nitric oxide synthase, and that can what that does is to produce radicals which kills infected cells, but it also can cause damage, immunopathology for sure. Does SARS-CoV-2 show any signs of antibody-dependent enhancement? No, there's only one serotype, right? So there shouldn't be any of that. Nope. Thank you, Barb Mack, for your contribution. Appreciate it. Would the use of antibodies for the other serotypes? No, because you'd have to diagnose it really early, right? Because by the time you get some symptoms, then it's too late to do anything, yeah. All right, folks, thank you all for coming today. Hope you learned some virology. I'll be back Wednesday. If not, if I don't see you Wednesday, have a great holiday. And as always, be safe. Take care. Bye-bye.